Thank you very much, Sarah. I'm sure there are some questions. We have 5 minutes for that. So, Vladimir, can you just start?
Okay, thank you. I just want to ask that, well, if for what you say optimal dragging, does this mean that conductance is maximized for your systems?
So we do optimize the work. So that's the functional that we optimize. Maybe one could say that optimizes conductance. I'm actually not sure. I would have to think about that. I'm not particularly sure how you would think about that in this context.
Okay. Thank you.
Thank you.
Okay, then we have David.
Nice talk, Sarah. I was curious about this last point you made where you optimized based on the symmetry and you found that you got the same solutions. Can you comment at all? Does the symmetry landscape work better for optimization? Have you thought at all about kind of comparing the speed with which they optimize?
Yeah, that's a very interesting point. So in the algorithm that we used here, which was a genetic algorithm, here you can actually see, so the red curve is optimization for symmetry and the black one is for work. And there you can see that it's not quite clear. I mean, they are more or less converging at the same speed, I would say. And also I played around with it, so it depends a lot on the parameters. So there's no general principle, I think, from that perspective. However, there are actually algorithms already, a lot of algorithms that work specifically on symmetries. So actually Megan Engel, I don't know if she's here, she told me about that. So I think it potentially opens the possibility to using other algorithms because the symmetry is a property of the function you optimize itself, right? It's different from optimizing a cost functional, which is kind of not directly related to the function itself. So somehow I could imagine that you can work with that. And also, I mean, you can definitely improve the speed immediately because if you know that you only optimize on the class of symmetric functions, you have a much smaller class of functions you search in. It's also really useful for analytical optimization because then, yeah, you can immediately start just looking at symmetric solutions. It's a much easier problem then. Thank you.
Thanks.
Nice. So Martin Rosenberg.
Yeah. You hear me?
Yes. Hi, Martin.
Hi, Sarah. Nice talk. According to you, the curve that you show, it seems that the jump at the initial or the final moment are smaller with the non-Markovian case.
Yes. Yeah.
Why?
So
why? Why? Do you know why?
Yeah, that's a good question. So that is we actually observe that all the time. The problem is a little bit with comparing them, that you cannot quite compare Markovian and non-Markovian case because you have different frictions, right? You need to think about whether you compare instantaneous friction or integrated friction over the whole time period. So I'm here comparing the total friction when I plot these two curves together. However, it seems that in general, I would say in general, the jumps seem smaller. Yeah, I'm not quite sure if I have a good answer to that. Other than maybe if you do a big jump, you also put a lot of energy into your viscoelastic surrounding because you displace the particle quite a lot, and maybe that's not something that you want. Maybe this is wasteful.
But would there be a way to make the jump disappear?
No, they are always there, so they are generic in our solutions, but they just become quite small. Yeah, thank you.
Okay, thank you.
So we're running a bit out of time. Maybe just a quick final question from Rui Cheng.
Hi, Sarah. Thank you for the very cool talk. So the symmetry of the solution relies on the symmetry of the quadratic potential. Is that correct?
Yeah.
So I was wondering, can this idea be slightly generalized? For instance, consider two harmonic potential which are stochastic switching, like for instance, there are two potential and the system is switching between these two configurations, or
Yeah, thanks. That's a great question. So I'm also thinking about generalizations these days quite a bit. I think in general, whenever you can write your work or another functional, it doesn't have to be the work. If the heat happens to have this property, for example, then you're also good. So whenever you can write it in this quadratic way, you would expect the symmetry. I'm not sure for the problem that you just mentioned. I would have to think about it, but I think it's nice to pose these questions, and that can give you insights into your approach just for these these kind of mathematical reasons without even any additional knowledge about the process. Thank you. Yeah, but I'm very interested if somebody has ideas for other processes that can fulfill these properties.
Sorry,
Yeah,
Gonzalo.
thank you. Thank
yeah,
you
thanks.
both.
Okay, so I think we're running out of time a bit. So I see also there are a couple of questions on the chat. Maybe Sarah can just try to answer those on the chat directly, or maybe later during the break. Should still
Yes,
have some
thanks.
time then. And then let's move on to our lightning talks. And our first speaker, we stay in Göttingen, will be Rick Bebon. So the
Yes.
stage is yours.
Can you see my slide?
Yes.
Perfectly. So thank you very much for the introduction and also for organizing this very nice workshop. So I will continue with talking about memory. Especially, I want to talk about a project we did together with two experimental groups. So, and also I want to start basically by highlighting these two collaborators of us. So, first I want to mention the group of Carsten Sönnigsen from the University of Mainz. And then also the other collaboration was done then with Thorsten Hugel and his group from the University of Freiburg. And here I want to especially highlight his two students, Leonie and Julia. And what they did is in the lab, they measure single proteins, and they are especially interested in the dynamics of the heat shock protein HSP90. And this is also from a biological context, a very interesting protein. So very simply put, it's a protein that helps other proteins to fold properly and also protects against misfolding. And as the name already suggests, it also protects them from external shocks, for example, heat shock. And not only from biological, but also from a physical perspective, it's a nice playground to look at different approaches. And this is because it exhibits this very large conformational changes. So here on the right-hand side, you see that this protein undergoes a rather stark conformational change from a V-shaped open conformation to a closed shape. And in their lab now, they can really track this open and closing motion by a technique that is called a plasmon ruler spectroscopy. And to put it in very simple terms, the idea here is that you attach two plasmonic nano gold particles to each of the monomers of this HSP90 dimer using some complicated biochemistry. Then due to the plasmon resonance, you can observe using a dark field microscopy, you observe a redshift in the wavelength and also a shift in the intensity. So essentially what they measure in the lab, or what they get as a result of the measurement, is a plasmon ruler signal, Q of t, which is a one-dimensional proxy of this precise open and closing motion of this protein. And here I show you a small snippet of one of these trajectories. So the actual trajectories are much longer, they're multiple hours long even, but even for this very short one, we already see these two distinct states. So here in the low plasmon signal, this corresponds to this open shape, and the high signal corresponds to this closed conformation. And some of the key questions we wanted to address now is, first, we wanted to look at, are there non-Markov effects? So is there memory present in these time series, since it's a one-dimensional projection of a very high-dimensional system? So does it have memory about hidden initial conditions? And if so, on what timescale does this memory emerge and also then decay, and maybe has it some biological relevance? And the key approach we wanted to employ is, of course, you can use this, or you can attack this problem from many different angles. And we employed a very simple and naive idea. So we wanted to directly check whether the Markov property is violated or not. And also you can come up with multiple ways to do that. And we especially looked at the Chapman-Kolmogorov equation, since you know, just to repeat it again, every Markov process obeys basically this Chapman-Kolmogorov equation, which is an identity for the transition probability, which I denote in my talk as this G here. So the probability that you start at some Q0 at time t0 to end up at a different Q after some time t. And this equation now gives you this identity here. So you can also, here the right-hand side shows that you can go to intermediates. So first you go to an intermediate Q prime at some time tau. And then from this intermediate, you go to the final point. And this integration is then simply the summation over all possible intermediates. And one further assumption we make is that we assume time homogeneous process, which we can also directly test from the measured data. And what we now want to check is if this equality is violated or not. So we introduce the right-hand side as this GCK. So this is our construct. This now also depends on tau. And we know for a Markov process, this is equal, and we now want to apply this equality for a process which we don't know if it's non-Markov or Markov. So the key idea is that we want to check are these two equal or not. So as I mentioned, we now compare these two quantities, and before we do that, we have to infer them. And the nice thing about this approach is that you can directly infer them from your measured trajectory. So you don't have to assume a specific model for the HSP90 dynamics or have to fit any parameters. And if you now want to infer a histogram, for example, like this at some specific time t prime, you can do it like this. So the key idea is you look at your trajectory. So here I show you the sum of the data points, for example. Then you specify some initial bin Q0. Then you look at all the data points within this bin. And then you essentially make a very simple histogram analysis. So you look where does the trajectory end up after your time t power. So for example, if I look at the blue data point here, then I choose some t prime, and after this t prime, the trajectory is now in this red bin here. So this data point would correspond or impact the value of this blue bin here. And then you repeat the whole machinery for all data points, and you end up with your inferred transition probability density G, and you repeat this also for the CK construct, only then the integration is basically a sum. So now we have inferred these two quantities, and now we want to compare them to make some statements about memory. And to make this a little bit more quantitative, we compare them using KL divergence. So very simple, we compute this. And this now then is also a function of your tau parameter and this initial bin you choose, and this as a function of time. And since it's a function of time, this also allows us to introduce what we call a memory timescale, which in this case is just the asymptotic exponential decay of our KL divergence. So this gives some information about the timescale on which the memory decays if we detect some. So and with this KL divergence, now essentially two different cases can appear. So the case number one would be that we would observe that we infer a KL divergence that is equal to 0. And this is only the case if and only if our two quantities are the same. So they obey the Chapman-Kolmogorov equation. But here the important thing is that this does now not imply that our time series is a Markov process, because there also exist non-Markov processes that fulfill the Chapman-Kolmogorov equation. And the second case now would be, which is also more interesting in our context to infer memory, is if the KL divergence now would be strictly larger than 0, which in turn implies that our inferred quantities are not equal. So the process we observed and computed these quantities from now does not obey the Chapman-Kolmogorov equation. And in this time, we can then conclusively say that our measured time series QT is a non-Markov process. Now, basically, I'm already coming to the punchline of my talk, which is we now apply this whole machinery and procedure for the experimentally measured time series of this heat shock protein HSP90. And these are some of the results. So let me guide you through this plot. So each panel, I show you this KL divergence as a function of time. And yeah, the first thing we observe is that, okay, we have a quantity that is larger than 0. So, okay, the first statement we can make is we detect clear signatures of non-Markov effects or memory of hidden initial conditions in our time series. Then the next thing is we can look at these memory timescale I introduced on the last slide, and here we found for all the different examples we looked at that for this protein, the timescale was found to almost 50 seconds. And the last thing is basically the two different colors. So the two different colors correspond to these different initial bins Q0. So the orange color corresponds to the closed conformation, and the blue color corresponds to this open conformation. So here we can probe the memory of hidden degrees of freedom basically in this open or closed conformation. And here we see that it appears that the closed conformation has more memory about hidden initial conditions of hidden degrees of freedom than the blue one. So with that, I would also then come to my conclusions already. So I hope I could convince and also motivate that if you want to analyze single molecule experiments or trajectories, it's important to look at memory effects and also to test whether non-Markov effects are present or not, especially in a context if you want to do some Markov modeling at a later step, if you want to, for example, study dynamics, then using Markov jump processes. And then in the specific example of this heat shock protein HSP90, we could confirm and also quantify memory, which was found to be present for up to 50 seconds, which you can argue for a single protein is could be quite some time. And then also lastly, I want to again highlight that for this analysis, it's important to remember in which way implications go and which way don't. So KL divergence of 0 does not imply that we have detected a Markov process, but each Markov process would imply a plot like this. And also the reverse, that a KL divergence larger than 0 implies a non-Markov process, but a non-Markov process does not necessarily imply a plot like this. So yeah, thanks for your attention, and I'm happy to take questions.